Hello, welcome to today's demola, and today we are going to be moving into topic 3, voice of the genome. We're going to start off with a crucial topic, cell structure, covering organelles, eukaryotic, prokaryotic cells. Let's get into it. Okay, so first thing we're going to do is we're going to talk about eukaryotic cells. So, important things to know, they have membrane-bound organelles. Yeah, the organelles is a structure inside of the cell that carries out a particular function, right? They have ones that are surrounded by membranes. Cool. They got ATS ribosomes. So we'll talk about ribosomes in a second. ATS is just the size. So just remember in an eukaryotic cell, ATS ribosome. DNA is linear, meaning it's wrapped by histone proteins, and it is also contained in the nucleus. So these are really important points to know about eukaryotic cells. So animal and plant cells are the main ones that you need to know the structure of. Right? And we've got a lot of different organelles inside of here. Let's just start outside, cell membrane. Right? So I'm going to highlight things as we go. So our cell membrane here on the outside, yeah, this just, it controls what can come into the cell, it controls what exits the cell, yeah. When we're looking in a cell, we've got our nucleus over here, yeah. So let's focus on the nucleus for a second. You'll see this black dot inside, that's called a nucleolus. It synthesizes ribosomes, but your nucleus as a whole controls the activity of the cell and it stores DNA. You'll see as well, we've got these gaps, right? These are called your nuclear pores. So your nucleus is surrounded by what we call an envelope, meaning a double membrane. Yeah, and that double membrane has gaps in it, nuclear pores, which allows things like mRNA to get out. We've got your rough endoplasmic reticulum, which I'm highlighting now. That is, it has ribosomes dotted on the outside, right? And that's mostly to do with protein synthesis. Smooth endoplasmic reticulum is lipid synthesis. That's over here. We got our mitochondria, which is to do with aerobic respiration and ATP synthesis. Lysosomes store enzymes. Golgi modifies and packages proteins. Centrioles are involved in mitosis, and ribosomes here are for protein synthesis, specifically translation. We do have all these definitions again, so don't worry too much. Plant cells, they pretty much do have everything in the animal cell except really the centriole. That's the only debatable one. But otherwise, it has everything else. But in addition, they've got chloroplast, set of photosynthesis. They've got a permanent vacuole, which is mostly about maintaining the cell's integrity, but also contains this thing that we call cell sap. If you want bonus points, the membrane surrounding it is called a tonoplast. You've also got your pits here, thinning of the cell wall, and a plasmodesmata, which is a gap in the cell wall. They carry out pretty much the same function. It's mostly about communicating between cells, so like transport synthesis, etc. Now, your plant cell also has this thing, a cell wall. It's an additional layer of security. And you need to know, very importantly, it contains this cellulose. Alright, cool. Here are all your definitions. So yeah, read through those. Get them down, memorize them. Very, very important. But yeah, just pause the video. Okay, so now we need to talk about prokaryotic cells. And now, very important thing to know, these are not the opposite of eukaryotic cells. They are different, yes, but they're not opposites. Now, they do not have membrane-bound organelles. So you won't find chloroplast, mitochondria, endoplasmic reticulum. You won't find those in a prokaryotic cell. They are not membrane-bound. What you will find is 70S ribosomes. So again, ribosome, we talked about a site of protein synthesis, but your 70S are just smaller. So you have smaller ribosomes in a prokaryotic cell, and now you have circular DNA. So it's not wrapped by histone proteins at all. It's not in a nucleus, it's circular DNA. Let's look at bacterial cell. So you'll see we've got labels again. Here's your circular DNA. Yeah, so you'll see that's not in the nucleus. Sometimes they'll call it saying it's in a nucleoid kind of thing, but really it's just floating in the cytoplasm. Cool. What else we have? We have a cell wall. Now this cell wall is made up of peptidoglycan. So same kind of thing in terms of protection structure, but this time it is made up of different sugar, peptidoglycan. Now what else do we have? We have plasmids. We're going to talk about its function. They can sound like extra genes. Yeah, so like antibiotic resistance, sometimes fluorescence, stuff like that. And the flagella is just this towel that lets it to move through fluids. And we also have your pili. This allows it to connect to other things. So you can see definitions, the important ones down here. So yeah, again, pause the video, get these down, memorize them. Very, very important to know. Okay, so we're kind of just going to look at this table very broadly. It's a very, very, very important table. Can't emphasize it enough. Now, why is it important? They love questions like this on the exam. They love asking you eukaryotic cells versus prokaryotic cells. Yeah, so just know these differences. We've talked about some of them we haven't. So the cell size, just know your eukaryotic cells are larger. So 10 to 100 micrometers versus 0 to 0.5 micrometers. If you don't remember the exact sizes, not the end of the world. but just know that eukaryotic cells are larger, they're more complex. We talked about nucleus, we talked about DNA, we talked about histones, we talked about organelles, we talked about ribosomes. Something else I wanted to mention, cell wall. Plants, we said cellulose. Fungi are also eukaryotic. They're also eukaryotic. Their cell wall is made up of chitin. Only remember that about fungi. You don't need to go and remember anything else about fungi cells. And we talked about bacteria being peptidoglycan. Cell structure as well, just want to emphasize. So your eukaryotic cells, they tend to be multicellular organisms, right? Organisms made up of a lot of cells, just like you and me. Some of them are unicellular. So think of an amoeba is an eukaryotic cell, that's unicellular. And your prokaryotic cells are almost always going to be these unicellular bacteria. Yeah. These organelles, again, pause if you need to, just have a look. Make sure you know that these organelles are not found in prokaryotic cells. So if that comes in an exam, none of these will be found in a prokaryotic cell. Cool. Now this is the last teaching bit of this video, but I just want you to go on a journey with me. So we're in an animal cell, right? We're going to start our journey here in the nucleus. Yeah, with that dot. Cool. When the nucleus, the big boss says, okay, it's time to read a gene, it's time to transcribe. Yeah? We should know transcription produces mRNA, this guy in blue. Alright? So, now we've become mRNA. Your mRNA is going to leave that nucleus with the nuclear pore. Cool. When it goes, it meets the rough endoplasmic reticulum with the ribosomes on the outside. So let's interact with the ribosome, like one of these guys here. When we interact with the ribosome, we are going to go through translation. We're going to form a primary structure protein, which is a sequence of amino acids joined by peptide bonds. Yeah? Cool. Then we're going to enter into our rough endoplasmic reticulum. And once we're there, we're going to undergo a lot of folding. So we might become a secondary structure protein, meaning we form hydrogen bonds in between different parts of the chain. We become an alpha helix or a beta-beta sheet. Then maybe if we're doing a bit more, we might end up becoming tertiary structure. So we might form ionic bonds, disulfide bridges, hydrogen bonds between those R groups, fold into a nice 3D tertiary structure. Either way, we have been folded, yeah, in your rough endoplasmic reticulum. And then we become this guy. So this is a secondary or tertiary structure protein. When it's there, it's going to make its move to the Golgi apparatus over here. It's going to make its way to the Golgi apparatus. Didn't mean to rub it out. We're going to make our way to the Golgi apparatus, and this is where we're going to be packaged or modified, right? So modification, that could be maybe I want to add more proteins. So there I've got one tertiary structure protein. Maybe I'll put in a second, a third, or fourth to form a quaternary protein. Yeah, multiple polypeptide chains. Or what I could also do is maybe I'm adding a glucose. Maybe I want to form a glycoprotein. Either way, modification and then packaging. So in the packaging, we might be put into one of these guys, which is an intracellular, could be like a lysosome, or we put in an orange guy, a secretory vesicle, right? And that secretory vesicle needs to secrete its stuff, right? So it's going to go to the cell membrane, fuse with it, exocytosis is going to release it all. Now, I just need to make sure that you know that process. So that's the visual one, this is the written. So you can see both at the same time. This gets asked so much in exams, which is why it's really important to know. We've undergone key topics like protein synthesis, exocytosis in this. So make sure you are, and protein structure. So make sure you're really happy with those. We do have videos on those on the channel. But yeah, that's the cell trafficking. Either read back, listen back to my explanation on the diagram or read below. Just make sure that you know this process. So that was the introduction to topic 3, where we're talking about cell structure. Now it's time for questions. So first question, you're comparing prokaryotic cells and eukaryotic cells. Then you're looking here, you need to identify, first of all, what the organelle is, and then talk about its function in terms of cell trafficking. And then finally, we've got an electron micrograph here. No, we have not talked about this, but it says it's a nucleus. Look at it, see if you can see the structures that mean that this is a nucleus. Okay, so it's time for the answers. Now, the first question, we've talked about this in so much detail. If you need to go back to the table, go back to the table, but you'll see it's common themes that we've talked about. We've talked about membrane bound versus non-membrane bound. We talked about plasmids, pili in prokaryotic cells. We didn't mention slime capsule. We'll talk about more when we get to tuberculosis in topic 5 or topic 6. We talked about 7CS versus 8CS ribosomes. We talked about endonucleus versus endocytoplasm. We talked about circular versus linear. We talked about peptidoglycan cell walls. Mirroring is the same thing. And I had to cut off the marching, but you would see like cellulose for plant cells as well. That was the first question. Second one, you need to identify. So you should be able to see A is your rough endoplasmic reticulum. You can see the ribosomes here. B is your Golgi. C is your secretory vesicle. So once you've got those, you need to know what they do. So the protein is going to enter the Golgi. when it enters the Golgi, like we said about modification, doesn't need a specific example, just modification. Then it's going to be put into the secretory vesicle, and that's going to be exocytosis. Cool. There was a harder one. So you can see the answers there. Let's spot these things on the electron micrograph. It's a bit peak because you wouldn't have been able to zoom in, but we can see here, that's a nucleolus, yeah? If they told you it's a nucleus and you see this massive dark structure inside, polynucleolus. Now, the nuclear pores are a bit hard to see, but I'm going to try and highlight some so you can see there are tiny, tiny gaps. Very hard to see, the tiny, tiny gaps. And then if you zoom in a lot, you can kind of see that it's a double membrane structure as well. See one side of the membrane and another there. So these things are hard to see, maybe except for the nucleolus. I would encourage you to just, if they told you it's a nucleus, you should be able to remember the kind of things you expect to see. You expect to see nuclear pores, you expect to see nucleolus, you expect to see a double membrane. I'd just mention them. Even if you didn't exactly see them, because like I said, hard to see, especially if you're on a computer screen. But guys, if you know it should be there, just mention it, because it's more about getting marks. So yeah. Okay, and now it's time for the A-star tips to round off the video. So, what do you want to do? Differences between prokaryotic cells, eukaryotic cells comes up so much, know it. Functions of the organelles, again, it seems really overwhelming at first when you're just flooded with all these different organelles, yeah? But I promise you it's not as daunting as it seems. Just keep on familiarizing yourself with it, it will come. But yeah, you do need to know about the functions of those. Drawing the organelles as well. So, for example, I've drawn all the organelles, you can see what I've drawn. You need to be able to confidently do that as well, or at least to identify them if you have an imaging exam or you have an electron micrograph. We haven't talked about electron micrographs too much in this video, but very, very important to know. Cell trafficking as well, also really, really important. They ask that question a lot, so make sure you're happy with that process. If you need a recap on protein structure, protein synthesis, or transport across membranes, go back to previous videos. We've got it all there. Now, what do we not do? A lot of people think that prokaryotic cells and acellular are the same. They're not, right? So prokaryotic cells are still living, so they're not acellular. They're still living. Also, fungi as eukaryotic cells, it's just commonly forgotten. Just remember, fungi are eukaryotic cells. And the cell wall contents, this is just what I'm talking about. Cellulose for plants, chitin for fungi, and peptidoglycan for bacteria. Just remember those, you'll be fine. So, I really hope that video has been helpful. If it has, please like and subscribe to the channel. If you have any questions about what I've said, just ask away in the comments below. I'll do my best to answer. We've also got a TikTok and an Instagram. TikTok and Instagram, both at A-star demola. Links will be in the description below. But that's all from me. Again, any questions, drop them in the comments below, and I'll catch you in the next one. Bye.